Okay.
All right. So,
Okay, go ahead.
all right. So, thank you, Amy, and the class for inviting me to this, to do this presentation. So, like Dr. Myrtle mentioned, this is a collaborative project that that Dr. Myrtle herself have been heavily involved for now actually 2 years. So, now, before actually getting into the project, let me talk about the background, sort of the premise for this particular project. So we are going to study this research that we are talking about is studying wildlife distribution along urban to rural gradients using sort of a automated monitoring method. So it's not an active sampling methods, rather a passive sampling method, which actually is better than active sampling. So what is an urban to rural gradient? So urban to rural, when you move from the urban core of a city towards and move away from the urban core, you actually get this urban to rural gradient. So here first you actually start with the urban center, right? So you pass then through the urban periphery, including the areas where you get neighborhood development, residential areas, and then you transition to variable degree of rural settlements where you see, certain level of agriculture, and then you transition into wild areas or wilderness where you actually largely get more open green space. So when you move, like what kind of environmental variation do you see? Human population density declines towards the rural end. The soil sealing, the impervious layers becomes less prevalent and more natural land cover opens up. The diversity of animals and plants becomes increasingly increases as you move towards rural end. But that is sort of a debatable question. I mean, is it like, can we universally accept it? It's an interesting concept, interesting theory to further explore. Air pollution predominates in the urban environment, becomes less of a problem in the rural environments. Water runoff and flood regulation, right? Natural flood regulation and natural surface runoff is more predominant in the rural areas. There is very little natural flood regulation in the urban environments, and usually, it's done by engineered structures. There are a lot more anthropogenically influenced green spaces in the urban center, largely like community gardens, urban agriculture, right? Those are like largely human driven, like urban parks. Those are largely those are natural open spaces or semi-natural green spaces that are largely structured but with human influence. And there are many things that is pollination. That's something actually Dr. Myrtle knows more than me, that could be actually also variable in this gradient. So now, a very interesting theory that we are trying to understand with this project is biotic homogenization. An increase in by definition, what is biotic homogenization? It's an increase in the spatial similarity or geographic similarity of a biological variable over time, which can be evaluated or which can be quantified by comparing those variables in a pairwise fashion between two different habitats. So biotic homogenization or this biological variables measured to assess biotic homogenization can be genetic, taxonomic, like species, functional. So homogenization can be broken into those. When I say functional, ecological functions, certain species play in their environment, like plants fixing nitrogen or insects performing pollination, big cats or carnivores performing a predation. Those are the functional elements. So before that one, like right over here, like, it's easy to explain this phenomena through a diagram. So regionally, like there are natural species pools, right? You have generalist species as well as specialist species. Generalist species actually have generalized ecological strategies, right? They can occupy different environments, can use different resources, can switch between different resources depending on the availability, right? Their niche requirements are very are not narrow, they are quite broad, so they can switch and pick as things are made available. Specialists are the opposite. They are functionally very specialized. They require very specific environmental conditions. If they are not met, they cannot have an alternative. So their fitness declines if those requirements are not met. Now, add our urban rural gradient, right? That creates an environmental filtering effect. So what happens because of that environmental filtering effects, when you actually have very high levels of anthropogenic influence, human influence, disturbances, urbanization driven ill effects, you tend to get more generalist species. On the opposite end of the gradient, where you look into rural environments, the filtering effect, the anthropogenic effect, urbanization's ill effects are less, so you get a lot more specialists, right? So so that's actually what this is the underlying biological principle in biotic homogenization, right? So what you do see is actually, if you look at the function species perform, right? As you move towards the rural end, you actually get more loss of functional diversity, right? Or rather more, not loss of functional diversity. As you move more towards rural environments, you get more functional diversity. So reduce functional homogenization. What do you mean by functional homogenization? Similarity, increasing similarity. So in the urban environments, most of the species that are left are functionally similar. So in the urban environments, you have high functional homogenization. Species left in the urban environments are functionally more similar. Now, another way to think about this is actually when urbanization creates an environmental modification, it triggers two things. One, triggers extinction or local extirpation of native species. On the other hand, it invites biological invasions, right? So both of them, right, not just this species loss or environmental filtering, but introduced species, invasive species, can also be a factor that you need to consider when you are theorizing or researching biotic homogenization. Now, this actually, I'm not going to go over this. This is like, just to iterate biotic homogenization in a different way. And the reason I bring this up is actually right here, the biotic homogenization example comes from deforestation or deforestation gradient, right? Or land degradation gradient. So although I apply biotic homogenization into the urbanization, that theory of biotic homogenization can be applied to any environmental gradient. Now, this particular discipline has a really interesting temporal trend with time, right? Earlier in 1950s, we barely started talking about it. In fact, you will not find many papers in the 1990s, less than 5 papers probably, that actually address this idea. I think the first paper, as I can remember, came around 1990, 1998. I think that's the time frame. I can remember the first paper being published. But the interesting scenario is actually, like for another 5 years, there's a slow pickup in terms of research interests, and then a tremendous increase and growing increase in publications emerging based on biotic homogenization. Right. So here, like, on y-axis, we are looking at time. On y-axis, we are looking at total number of papers, articles quantifying biotic homogenization. Right. And then on the other hand, on the flip side, you are looking at the number of articles citing biotic homogenization. So we kind of interestingly see, publication trend and the citation trend follows very closely. So it's interesting to see that us in the scientific community are interested in studying this phenomena. the pointer back. And also look at global trends. It's really interesting to see that. Largely in the the larger greater Europe, right, and the northern Asia, there was a this principle has been extensively studied, or theoretically, or in from the empirical sense. Then in North America, right? But if you look at, unfortunately, the southern hemisphere has not studied this very well, right? So we are trying to expand what we do into the southern hemisphere, but that does not come easily. So now, specifically to our research network, this research project was initiated by Lincoln Park Zoo in Illinois. We actually right now have about over 30 partners collaborating in this project. That list is growing, and in fact, our map you see in this corner is grossly out of date because it doesn't even include us. We should have a dot over here. I perhaps this is actually way about time for me to update these things.
Yeah, you're done. Yeah.
I know it's been a while since I actually thought about. I do need to do this, Amy, before the talk that is coming up in the Zoo New England, right?
Right.
And then now, since we have a lot of data, I should actually add that data. But I always wanted to create the theoretical foundation first, particularly for your class, because some of them may not be familiar with the theory, but that's something you and I need to talk about for future classes. So and then we are studying urban-rural gradient in this research in multiple cities. So each partner we are talking about represents a single city. So Dr. Myrtle and myself represent Greater Boston area. And so our gradient actually represents urban-rural areas, forested areas, exurban areas. Those exurban is the areas that you find between urban and rural. And then residential areas, right? Areas that are actively managed, including some farmlands, like sort of former farmlands that are no longer actively being farmed. In our transect, we have precisely 27 trail cameras. Our network requires somewhere between 20 to 25. Some of our partners actually have over or as many as 30. And certain partners actually have over 100. I don't know how they manage that much work. I wish I could. Probably we don't have that many green space to put, Amy.
Yeah, right.
100 cameras. Yeah, no. Those partners who have about 100 cameras, they are in the Midwest. So now we don't have that much green space left. So each camera in our transect are about 1 kilometer apart from each other, and they're activated. The data collection happens for a minimum of 4 months in a given year, representing the seasons, the 4 seasons that we are interested in, because we wanted to know not only the occupancy of wildlife, but how that occupancy vary across different seasons. So earlier we were actually thinking about sort of adding a lure, but we are realizing the lure effect is actually hit or a miss. It could be more confounding factor than a helpful factor. So we actually, after some experimentation, even before I formally set it up, I decided not to include the lure, but I wanted to mention that because if you guys are doing particular study, lure might be helpful, but it depends on what specific location, what species you are studying. So this is, although I have only one dot in Canada, we technically have 2 partners in Canada, and we are we have a partner from Germany, we have a partner from hopefully from South Africa, and unfortunately we are still unable to get any partnerships from Mexico. That would have been wonderful. Anywhere in Latin America might be a wonderful addition, but we don't have it unfortunately, or from Caribbean islands. So this network, although my involvement is rather scientific and partly educational, educational from the educational perspective, it's largely mentoring student towards research. But like I show in this diagram, it grows, it can go beyond that, right? And we are actually trying to seek other things we can do with this kind of data generation, going towards not just theoretical ecological research, but also going towards applied applications, developing applications that help better land management. That is something we are still interested in. But unfortunately, it's hard to keep your fingers in all the possible jars, although you wanted to. Well, it's a struggle always, us faculty members and scientists and actual practitioners That communication is a little bit hard. Now, to talk about specifically our data transect, you are on the map you are looking at specifically the 27 locations where we have trail camera deployments. And this is specifically, like Cambridge area where we have actually 5 different cameras. I do not know whether Dr. Myrtle's the lab work that you guys going to do. Well, actually, I think I know. It's going to be centered in these areas. You are looking at 2 sites in the Fresh Pond. It's a Cambridge city property. And then Belmont, there is in a Macedonian sanctuary that Dr. Richardson has been working with us to get that done. And then we have one on one of your southern campuses and Mount Auburn Cemetery. So those are the locations that probably you guys will be visiting with Dr. Myrtle for this particular project. So hold on, Amy. I'm on a call. Yeah, it's kind of weird, like, people knock on your door, although I have like do not knock. It's
Yeah, that's okay.
sorry about that. So this is actually the so I mentioned earlier the partnership I have with Lesley University and a couple of faculty members. But it grows beyond that. We partner with the city. We partner with City of Cambridge, City of Boston, several other land trusts. So this is a Zoo New England. So this is a Arnold Arboretum. This is a big partnership, right? And right now we are working on a couple of other grants. Well, actually one grant, and hopefully this efforts will actually magnify in future or continue at least in future. So this, so far as we are aware, this is one of the largest international research network that we are aware of that targets urban wildlife research, right? Not any biodiversity, urban wildlife research. So we are not married to universities. We encourage participation from all the sort of organizations that are interested in urban wildlife. Yeah, not just for students and faculty. I mean, like I mentioned, we want a lot of other practitioners, policymakers involved in this kind of work. And we generate so much data that is largely useful for generating publications. For example, Dr. Myrtle had an intern almost for a year earlier, right, helping us with this research. And then I had a student who did an honors thesis based on this. And then we are currently doing more analysis on her thesis work to get it published. And we already presented some of our research at a conference, and Dr. Myrtle was a co-author. Dr. Myrtle and Dr. Richardson both were co-authors of that conference. So this can be heavily, I mean, I'm actively using this for both graduate and undergraduate research, and hence that's why actually, we're doing this presentation to the class. And there are lots of, I mean, this kind of work is cool and catchy, so you get a lot of attention when you get connected to this line of work. And ecological questions you can answer, like, I mean, they are very plentiful, right? So you're studying your urban-rural gradient. So any differences in occupancy patterns of predators, of prey species, vectors, right? Those are like some interesting questions you can answer, right? And then what kind of environmental variables are actually driving at local to landscape scale when it comes to wildlife occupancy? And how their behavior is affected based on lunar cycle, right? And another project that we are currently still working on, the data analysis in this kind of work takes so much time, right? Almost like a couple of years. It takes a couple of years to collect enough data and another couple of years to analyze data. So these are like really, analysis heavy, cyber infrastructure dependent research projects. But the way you can contribute is so, plentiful. And so the nightlight effect is another one, right? Like, because in urban environment you have urban nightlife, in rural environments you don't have it. So does that change the detection or activity of wildlife in rural versus urban environments? And generally, like going back to the biotic homogenization theory, how much of species loss do you experience in urban environments compared to rural environments, right? And then does the type of urban green space matter? I mean, the green spaces we study, some of them are conservation dedicated, some of them are recreation dedicated, some of them are education dedicated. So some of them are hunting dedicated. So does it matter how you manage the urban green space? So there are so many questions that you can address, right? So I'm not going to go over this one, because Dr. Myrtle will explain how you are contributing as a class to this particular project. But I do want to mention, Amy, you might actually want to correct me after I mention this, or to the class. Anyone who's interested in tagging wildlife, right? That opportunity exists. It is as simple as sitting in front of your computer, opening our data portal, and tagging what species you see. In fact, Amy, I don't know whether you heard this, during summer, 2 interns with the not arboretum, the cemetery, Mount Auburn Cemetery, they actually volunteered to do photo tagging.
Oh, okay.
Yeah, I
Great.
can't. Gosh, Lena was
Yeah, and
one.
Daphne, I think.
And Daphne, yeah, there you go. Those two. They did a wonderful job and they enjoyed it. So anyone who wanted to do this, I welcome that, and you can contact me through Dr. Myrtle, Professor Myrtle, and I'm happy to help you out and assist you to be a partner in this project. Because, and in fact, it's kind of, I'm jealous of those who get to do photo tagging, because I am the data analyst, I'm the statistician, right, who sits behind the computer and do the programming, math, and statistics, and paper writing. Okay, my students and any student who's doing the photo tagging have the fun time seeing really weird stuff happening, like, I mean, like predation, like fighting, like, eavesdropping, to getting laid. I mean, you name it, like, being the third wheel. So I mean, I never get to see any fun stuff like this.
Is that from
And
one of our cameras, the raccoons there?
no, some of them could be like the previous slide. Some of them are at least ours.
Okay.
These are not actually, because that one. This is actually, I think, the fox squirrel. We don't have them here. We have the red squirrel. For example, I have never seen a red squirrel myself in our trail camera data. My students have seen it, right? And then Daphne and Lane both saw deer in Mount Auburn, and it's not supposed to be that way.
Oh, okay.
So like there are cool things we are finding out, and my students saw actually, coyote pups playing. I never seen that. I mean, there are like, and then my students saw aggression between a European gray squirrel and a red squirrel. I never seen that. So
Oh,
like
really?
yeah, like, like sort of aggressive behavior between a chipmunk and a gray squirrel. Like, you never see this. Fox chasing after a gray squirrel, red squirrel, like, those kind of things. Like, it's amazing what you see in these photos. And it, I kind of am jealous that I don't get to enjoy that kind of a cool stuff, like, and my students do, or the interns do. So generally, like, again, just to wrap this up, this kind of research, when it happens, like, we are doing it right now at city scale, but the same approach we within our research network try to replicate, not just within the city, but between cities, within a given ecoregion, and then between different ecoregions, and hopefully one day between different continents, because we do share species between continents. So the sort of the scientific appeal of this kind of research is fascinating, right? And it's also interesting to see when you compare, for example, across different cities, right? In this map, these y-axis, x-axis are different cities. Texas, Iowa City, Illinois, Indiana, Indianapolis, Kansas City, Denver, Colorado, and Fort Collins, Colorado, right? So you can actually see the darker color for a particular species, the more active that particular species is. And it's not surprising that you can see raccoons being like sort of the predominantly very active across different cities, across different ecoregions, right? And also like kind of a surprise in Iowa City, fox squirrel is one of the most active species, right? So there are really interesting surprise stories emerging when you look at these kind of a cross-city comparison. So this is actually one of the papers we recently published, and there are multiple work that we actually are currently in the process of writing, going towards analyzing the data. But hopefully within this year, we should have another paper from our city, the work that I and Dr. Myrtle do, that looks into our own city's data. Well, with that, I will actually open up for questions if we have time.
Yeah, sure, definitely. Yeah, make this full screen so we can see you. Give you a round of applause. Thank you. Does anyone have any questions?
Somebody is raising their hand. Alec?
Oh, sorry, go ahead.
I was clapping, sorry.
Oh, clapping. Okay,
Oh,
thanks.
that was a clap, not a raised hand.
Yeah, go ahead. Should be able to, yeah.
How did you come about this project for the wildlife?
I didn't hear the question clearly. If you want to repeat that, Amy, since you are close to the mic.
Yeah, how did you come up? How did you kind of get started with this project? Like, where did the idea come from, or how did it get started? Okay.
Yeah, so like I mentioned, that this is a research network. So I met the network coordinators at a conference, in the Wildlife Society conference, and that's actually how they recruited me. They saw my line of work, and the fact that my school is closer to the city. They actually sort of brought this idea to me, and I was also interested in this collaborative network research. And then I applied for a small grant to get equipment, and that's how I started rolling this idea. That's how this evolved.
Would you mind, I don't know if you have any, like, can you talk anything about anything that has been found so far in, I mean, you mentioned the paper on COVID wildlife. I don't know what that paper showed, or just any other interesting findings that might be able to share.
Yeah, so I mean, in my student's thesis, which I think, Amy, you were there in her defense.
Yes.
So she actually found out that for eastern gray squirrel, there is no impact of the impervious cover, land cover. Like, I mean, they remain active even at extremely low, right? There was the threshold from 100% occupancy of eastern gray squirrel, that dropping into somewhere around 70% happens so quickly. There was a precipitous drop in occupancy, but after that drop, there is almost no remarkable decline in the occupancy with increasing urbanization. I thought that was quite remarkable, because 60% occupancy, 70% occupancy, that is still high. That is quite high, right? And also another interesting fact is that we tested so many other variables trying to understand what drives the occupancy, other than urbanization, and of course the temporal effect. The seasonal effect is extremely important, and that seasonal effect is not the same between the rural end to urban end, right? So on the same day of a given sampling season, the impact on that weather, like colder weather or warmer weather, like it's not the same between, from Bridgewater or Halifax in the rural end to Cambridge in the rural, in the urban end. So that was also really surprising, on the same day of, like, I mean, it could be the same season, but the species occupancy is not the same, right? We are looking at the same species even, but it's not this, their detection is not the same between urban to rural environment. And coyotes, despite the fact that they have successfully invaded the eastern part of North America, from the Central Plains area, they actually are quite sensitive to urban development, right? They have a very low threshold for loss of occupancy, not loss of occupancy, for declining in occupancy with increasing urbanization. So that I think that was a really interesting finding from Boston data. And the thing I was going to say earlier was actually other variables, other land cover variables we tried: wetland cover, building density, human population density. We tried tree canopy cover, like none of those were significant predictors in driving species occupancy. It's just the impervious cover, which is a proxy of urbanization, which is so seems like that is the best proxy of urbanization, impervious cover, right? So and when I actually dive deeper into that paper, we will actually have a lot more interesting stories to share. Yeah, so those are some key findings that I wanted to mention.
Okay. Could you explain when you say occupancy, could you explain what that means in terms of the data? Because we'll be looking at that too.
Oh, good question. So when you set up a camera, right, and let it self-trigger with motion or thermal sensitivity, either you get a particular species as a photograph, which indicates presence, or you don't get a photograph of species presence. So it's 1 and 0, right? Now, but actually what happens is, simply because a particular camera does not capture a photograph of a particular species does not necessarily mean that particular species was not there, right? What it means is the species was not detected, right? There are many instances, regardless of the sampling method, a species could be present in a habitat, but it is not detected. Maybe we are not sampling the right time. Maybe our sampling method is not effective enough to capture all the individuals, or the actual population size could be so low that you don't detect them. So because there are so many sampling errors, just like I explained, you cannot analyze the presence absent data as is. You need to convert that presence absence data, the ones and zeros, into a probability of a particular species being occupied in that particular site based on the environmental variables and the temporal variables you collect, or that pertains to that particular site, right? So so in order to do that, you actually go towards very very complex arrays of modeling based on open source software and manually programming these things, right? So so doing that, so correcting for these imperfections in detection and converting presence absent data into a probability of a particular species occupying a particular site of interest for the survey is called occupancy modeling.
Okay.
So that's a that's a field that actually sort of has progressed fairly recently. It first started, we first started talking about this, like in 1980s, right? I mean, you guys are not even born when that happened, right? So except me and Dr. Merle, right? We are the two only people who were born when this actually happened. And then, like it kind of lost its touch for a while. And then recently, especially with the federal government, USGS involvement, to fund free and open access software, this became a really new interest. So there are lots of interesting models that was produced within the last decade or so Still, it's a very fast evolving thing. Another reason why it takes so much time to get this publication out is because, like I had to try several line of modeling. My student tried several line of modeling. And while I really enjoyed training an undergraduate to do computer modeling, it was not easy for the student to learn so much, like a graduate student level stuff in her senior year. But she handled it quite well. So yeah, that's actually, that's what occupancy modeling is.
Okay. Yeah, thank you. Just to clarify that.
Yeah, of course. I should have clarified that earlier on. Sometimes I forget.
Thank you. Does anyone else have any questions? I don't see any more questions. So, well, thank you so much for joining us and giving us the overview. And then we're gonna be setting up the cameras this week. So
Perfect.
see those in action, and we're gonna look at the data that we collect later on this semester.
Perfect, Amy. Thank you.
Yeah, thank you so much. Thanks for joining us.
Bye-bye.
Bye. Bye. Oh, so I'm gonna share something. Screen with everyone. Okay. All right, so that was kind of the overview for the lab that we're gonna start today, but it'll be long-term lab. We're gonna collect the data during the month of October, and then we're gonna do, once we've collected the data, we're gonna do some of what Lena was talking about. We'll be looking at the photos and storing the photos and seeing what different species we see in different areas, and then doing a lab write-up based on that. So today's kind of our first day of setting that up, but we want to actually do the write-up part after October. So I was going to talk a little bit as well about the lab part for that. Get that open. So this is, I'm not going to show you this whole presentation, so some of it repeats. I think that Helena was already talking about, but this is a presentation that myself, sorry. I'm just trying to figure out how to move. There we go. Okay. Moving out of the way so you can see. So myself, Dr. Richardson, and Javiel, does anyone know Javiel? He has been here last year. Javiel. He worked with us for this. He did an internship with us for this project all last year. So he was one of the people that Lena was talking about. He spent a lot of time looking at the photos and scoring them. So we did this little presentation at Community of Scholars last year. So let's talk a little bit more about our particular camera that we are working with. And I'll just kind of use this to introduce the lab that we're going to be doing. So this is just this overview. We already kind of talked about this. This you got already. So this big network. And one thing that's really exciting, I think, of being a small partner in this big network, that for if it's useful for you in the future, we also have access to data that's being collected in all the rest of the network, right? So, for example, if you wanted to do an internship or a capstone project and you wanted to compare data from animals in Boston to what's happening in Colorado or Texas or these other locations. Since we're part of the network, we have access to that data now. So it opens up all this data for potential research projects. So I'm not going to use all that in this class, but just something you can keep in mind for the future. If you are interested in this kind of thing, you can get more involved later on. Like people to know that we have that happening. So these are you saw this larger map already of the Boston network. And then this smaller box, these are the cameras that we're actually going to be working with. So we have 5 cameras in kind of the Cambridge area. One of them is on the Leslie South campus. So today, after this, we're going to walk over there and we're going to set up one on the South campus. You can kind of see what the setup looks like, what the cameras look like, how they work. I hope that I could get a van for today and we could do South campus and then Fresh Pond, but that didn't work out. So we're going to do South campus today. And Wednesday, when we go to Mount Auburn, we'll set up the Mount Auburn one there. If I can get a van for Wednesday and we have enough time, we might do the Fresh Pond ones as well, but if not, I'll just do those on my own at a time, just because partially the van thing took much longer than I thought to get together. So I don't know. I'll be looking at it for Wednesday. We will have it for later in the semester, though, fingers crossed. I'll talk more about that in a minute. So anyway, so today we'll set up the South Campus one at least, so at least everyone can see that will get set up and understand how that one works. And then there's one in Belmont, the habitat, Chris usually sets that one up. That's kind of on his way home, so he's gonna do that one. We don't have to be out to Belmont, so we're gonna set up the other 4. So the data is collected 4 times a year, like Felina mentioned. So we're gonna do the October one. So the data we're collecting, we'll use it for our lab, but it's also gonna go into the larger network. So we're, collecting official data that will be for the larger network as well. And these are just some fun photos. So it's fun to show photos. These are some of the different species that we've seen so far, just in our Cambridge cameras. I don't know if anyone, you can look at there if you recognize any of the species on there that look familiar. See when you recognize, you can yell it out. The deer, yes, that one is from Fresh Pond, I think. There is a possum, yeah, the lower left, that's a possum.
Skunk.
The skunk, yeah, in the middle we got a skunk. I forget where that one's from. That might actually be from the Leslie one. It looks very lawny.
That is turkey.
The turkey, yeah, we got a turkey over on the left. I think that one's from the Leslie one too. What?
With a swan.
Now what the next bird, that's a great blue heron. That was from Fresh Pond. There's 3 other ones. Anyone recommend the other 3? Yeah, top right, that's a woodchuck. That one's from Mount Auburn.
The raccoon.
Yeah, raccoon in the middle. One more, this one here. Get that one. The coyote, yeah, that's a coyote. That's from Mount Auburn Cemetery. It's like William was saying, there are some coyotes. They like Mount Auburn, they like Fresh Pond. They don't really like the more urban areas, but we found a pretty interesting amount of species. This is just kind of an overview of all the different species that we found. We'll see whether the stuff that we find in October, we'll be kind of replicating or kind of expanding on this data with stuff we collect in October to see if we see similar different ranges of species, different kinds of species. But this is from we've only had 1 full year of data at this point, because we just started, I think we started in the winter of 2020, or it might have been spring 2021, I can't remember. Somewhere in there. When we did this presentation, we only had 1 full year of data, so now we'll have a 2nd year. You can see things like gray squirrels and humans are extremely common. You see them everywhere. They have other things that are more specific, like a fisher. We've seen one fisher somehow on the South Campus. Otherwise, you haven't seen any of those. And you have some things like weasels or woodchucks, foxes. We've only seen in Mount Auburn. There's been some sites have species that others don't. So there's some things that are common to all the different sites. And the rest of this is our species accumulation curve, like we were talking about last time. We took those samples and we did a curve to kind of see how good we're doing at sampling. So this is pretty nice to see that's having done, this is based on using the months for the sites as different samples. At this point, looks like our community is pretty well sampled. We probably aren't a lot of species out there that we haven't found yet. But like Lena said, there could be species that are out there, but we're not capturing them because they just don't happen to walk by the areas that we have the cameras, or they don't utilize that habitat. So you can't necessarily say you've seen everything, because there could be things that just are very uncommon to find in the cameras because they're particular habitats. But based on the sort of species that you can commonly find with the cameras, we are getting a pretty good sampling from this, based on this data. And then this just has some, we're not going to see seasonal data. Obviously, we're only going to look at October data, but we have seen some seasonal patterns, again, just from our first year, where the bird activity was a little lower in the winter. The coyote activity actually increased in the winter, though, maybe because there's less humans out at that time. The squirrel activity was highest in the winter, which makes sense because that's their breeding season, so they're kind of already trying to mate with each other. So it makes sense that they were particularly high then. Deer activity was higher in the summer, which is their breeding season. So again, that makes sense. Which also means there could be more potential interactions with humans during the summer, and that has some applications for disease spread and things, because there are some diseases like Lyme disease that can kind of go back and forth. But I think that's, just future directions. Want to continue. I have 1 year of data, so we want to kind of continue collecting this data every year. We do have internships running in both fall and spring. If students want to do an internship as part of this project, we'd be helping with setting up the cameras, collecting the cameras, downloading the data, storing the data, analyzing the data, that kind of thing, and involve more classes in the project. Kind of these repeatable class projects, like we're doing in this class. Make this a long-term project for the ecology class that over over years, every semester, we kind of have a database of how things are changing over time. So up there, the other part is Marcus's part, so I'm gonna give that part. Basically, what we're doing in our lab, like I said, we'll set them up for October. Bring those photos back in the lab sometime in November and do some scoring. So we'll go through them, see what's in there. And we'll do some analysis of what species are at these different sites. You also have some opportunities to kind of think about particular questions that you might want to ask the data. So, thinking about what the differences between these sites. We're going to visit at least 2 of them. But I visit Rush Pond, we've been there before. There are some habitat differences between these sites in terms of how much green space there is, how much human interaction there is. So you might think about what expectations you might have for what species are in different places in the fall season. Differences between the sites. So we'll have a little bit of open-endedness where you can also think of some of the ideas that you want to look at with the data. But obviously, we're just looking at 1 month, so it'll be a little bit restricted in terms of how much you can do. But something that maybe would strike your fancy and you might want to look at for in the future on your own. Okay. Okay. I think it is all. I just have these little labs when I hand back to you guys. And actually, so wait, before I do that, the other thing I want to talk about is our Wednesday lab. Like I said, we're going to go to Mount Auburn. And I mean, thinking if I could get a van, I'm not super helpful because you're not getting back to me, and it's like 2 days from now. The whole van process is like new and confusing, and staff is leaving, and like, I'm gonna try. If I can get a van, then we'll take the van. But if we can't get a van, then we're gonna take the T. We'll basically just take the bus over here to Mount Auburn. I do want to ask if I don't know if anyone uses a car or driving here, that you want to drive and meet us over there, or they would be willing to drive other people. I don't know if anyone's in a situation where we take the T anyway. Drive here. You drive here, okay.
I'll probably drive the other school.
All right, so you have a couple people driving. Okay, so you might. How many people would you be able to bring with you if we
Probably got it.
just 2 people? Okay. 3, okay, so we probably couldn't all go in the car, then we have to take that subway either way. That was kind of my other question, whether it was possible to kind of organize.
If we do take the T, do we have to pay for it like separately?
Yeah, we have to pay for the cards. Yeah. I mean, I can bring a card that has extra money on it if people don't have bus cards that they can use. We take the bus, you take probably the 70, well, either we walk or take the 77 down to Harvard, and the 71 or 73 goes up to Mount Auburn. Kind of the 2 bus, a bit of a hike, cutting to our time, certainly, but we can still do it. We'll still have enough time to do the lab. Yeah.
So we're gonna meet here and then take the bus there, or we're gonna meet at Mount Auburn.
You can do it either way. I think that would be kind of the option. If I don't have a van, then it's up to people if you'd rather strictly at Mount Auburn, you could, or we can meet here and go over it together. I guess I'd probably just see who wants to do which one. And I'll be leaving from here either way, so I'm happy to meet people here and over. And then if you're meeting us there, I guess I just have to kind of time it to figure out what time we're going to get there, so I know what time I have to meet us there at. But I can send an email about that probably tomorrow. But if I haven't heard back for the van by tomorrow, then I'm giving my time. Maybe I'll do. Maybe you think about that now. Like, I don't know where you're going to be, but if you need you to be us there, if it's here, if we're not, we don't have a van to use. So kind of think about that for now, I guess. And we're going to be doing 2 labs over there. We'll be doing, we're doing 1 lab, which basically the aquatic sampling. Just while we're over there, we'll also put up the camera. That isn't really a lab, that's just putting up the camera. But the main lab will be sampling aquatic invertebrates, which is a lot of fun. Find some cool stuff in there. So that's it does involve some equipment, which is why it's nice to have the van, so we don't have to carry that. But we can carry it. All right, any questions on that? Yeah, I'll send out email by tomorrow. By tomorrow afternoon. We don't have the van, I'll just tell you. Meet here at this time or meet at Auburn at this time. Just kind of estimate how long it takes to get over there. And then probably ask you, let me know if you're not going to meet me here, let me know, just so I'm not waiting for me. I'll be over there. There's plenty of parking over there, so if you are driving, there's lots of places to park, so not an issue. Okay, all right, so I'll hand out back these little labs. So Alec, we are gonna take a quick break, and then we're gonna go go out and set up the cameras, so won't have to join us anymore. Well,
Okay, thank you.
all right, so hopefully we'll be able to see you again on Wednesday.
Yes.
Okay.